Hello and welcome everyone. Thank you so much for joining us for a very exciting overview of why stem cells offer the greatest potential we've ever known to cure disease. And along the way, we'll explain why we do what we do at the NYSCF Research Institute. I'm Susan Solomon, the CEO of NYSCF and founder. And today's webinar is intended to really give you an overall kind of basic of lay of the land of why stem cells are uniquely powerful and how we leverage this power to accelerate research into diseases that we all care about tremendously, like Alzheimer's, autism, macular degeneration, cancer, diabetes, and many more. We're also going to share how we leverage stem cells and automation to scale research to representative and diverse populations. I can't emphasize how important that is, and to catalyze new fields, combining cutting edge technologies like machine learning and gene engineering, which we also do. So in today's webinar, we really are only able to touch on a small portion of everything we do at NYSCF. And by all means, if you're interested in learning more about anything we discussed today, or you have a specific disease interest, please reach out via our website or through the chat function during the talk. Before we dive in, I want to acknowledge the transformative partnership of the Stavros Niarchos Foundation in making events like today's talk possible and in encouraging others to support our efforts. If you're interested in making a gift towards our education and outreach programs, it would be very welcome, and the SNF Foundation will generously match your gift one-to-one. So we hope that you will consider making a gift and doubling your impact with a gift today. So please visit the link in the chat for more details. And to all of our visionary partners who are watching today, we are deeply grateful to you. Our research and discoveries continue to be fueled by your philanthropic support. For those of you who are new to NYSCF, we look forward to sharing more of our work with you and hope that you will join us in our mission of accelerating cures for patients everywhere. It gives me great pleasure now to introduce Dr. Scott Noggle, NYSCF's Senior Vice President of Research. A longtime colleague and friend, Scott is an expert stem cell biologist with decades of experience in the field. He has been at NYSCF since our earlier days, guiding our research strategy, building our laboratories, and playing a core role in the research institute that we have today. Scott also played a critical role when we were conceptualizing and developing our unique robotic platform technology to create large-scale induced pluripotent stem cells and specific cell types. We call it the NYSCF Global Stem Cell Array. Scott has been a key partner in numerous exciting studies and research partnerships, including the recent ROSMAP study, which he'll mention, with Harvard Medical School and Brigham and Women's Hospital, which showed that stem cell models reflect the intricacies of an Alzheimer's brain. The ASAP Consortium, Aligning Science Across Parkinson's, which studies risk factors in the disease with scientists from Memorial Sloan Kettering, Brigham and Women's, University of Illinois, and the Garvin Institute of Medical Research. And our very novel PTSD work with Dr. Rachel Yehuda at Mount Sinai School of Medicine, to name just a few notable contributions. Currently, Scott leads our Alzheimer's disease research, which was also one of our very first disease research programs because of the unique opportunity that stem cells offered to give us a glimpse inside a living human brain. And Scott will share more about that shortly. Scott is the perfect person to walk us from stem cell basics through our cutting edge disease research, cell therapy, and drug discovery approaches. Scott, welcome.
Thank you, Susan. All right, so let's get started. Today we want to really take you through and give you a good understanding of what stem cells are, why we're excited about them, why we think they're the future of medicine. And as Susan said, our mission here at NYSCF has been to accelerate cures for all of our major diseases through stem cell research. And we focus specifically on using stem cells as a path forward to finding cures and treatments for many, many different diseases. So today, what I want to take you through is basically what stem cells are, what makes them unique and uniquely powerful. We want to learn a little bit about how we're then using those stem cells to teach us about different diseases, human diseases. And then how stem cells are paving the way for actually developing new treatments and cures. So I hope by the end of the discussion today, you'll have a good appreciation for just how exciting we see stem cells as the future of medical therapies. So where do stem cells come from? Stem cells, they reside in many different tissues of our body. During development, we go through stages of development that involve stem and progenitor cells that create all of the different tissues in our body. And those cells eventually generate tissues, and in those tissues are different types of stem cells. And so there's a lot of different ways that stem cells, sort of operate over time to create our bodies. But we find them to be very unique in certain characteristics that really make them well tailored for trying to understand the biology of us. How do we work? How do our bodies operate? How do they succumb to disease? So stem cells, we call them the superheroes of the body. They're unique cells that can become any cell type in the body. And, we have focused here at NYSCF on a particular type of stem cell called a pluripotent stem cell. And this is a cell that can make all the different cell types in the body, from brain cells to heart cells, blood cells, lung cells, muscle cells, all of the different cells that make up our different tissues in our body. But they have one really important other characteristic, and that is that they can self-renew. They can make an unlimited number of copies of themselves if you give them the right signals. We could fill up this entire room with stem cells if we tell them to keep forming more of themselves. And each of those cells that we would end up with would have the same capacity to differentiate and develop into all of these different cell types in the body. So we like these pluripotent stem cells, and they're a little bit different from the stem cells that end up in your different tissues in your body. Those cells, for example, blood cells, or the stem cells in muscles, or stem cells in the heart, they're really only capable of generating or creating more cells of that type of tissue. The type of cells that we're studying here, these pluripotent stem cells, are capable of becoming all the cell types in the body. And so we find them really, really powerful in that aspect. And we originally, started deriving these stem cells and making them stem cells from the early embryo. But over the past 10, 15 years, a new technique has come on the scene that's enabled us to create a very similar type of stem cell, these pluripotent stem cells, but starting with adult skin or blood cells, or many other different cell types in the adult patient. These cells have the same properties as the embryonic stem cells. They can make more of themselves, copies of themselves. They can also turn into all of the different cell types in the body. And it uses a sort of a genetic trick that tricks and pushes those adult cells to revert back to something that looks very much like an embryonic stem cell. And this was, recognized with the Nobel Prize. Shinya Yamanaka was the professor that first discovered these factors, these genetic factors that can convert an adult cell back into a pluripotent stem cell. a cell that can, make more of itself and also generate all the different cell types that we're interested in when we're trying to study and understand and potentially treat disease. And the advantage that this method has is that we're taking it directly from a patient that we're interested in. I can make a stem cell from myself. I can make a stem cell from anybody on the webinar today, and that stem cell will capture your genetics. That it will capture you as represented by that stem cell. So this has many advantages of trying to understand how diseases work. We can start from people who have particular diseases, say Alzheimer's or Parkinson's disease or diabetes. We know that that stem cell came from a person that has that disease. That makes them useful research tools for trying to understand those diseases in the long run. And we'll get into the details of that in a little while. But that also means that if we were to transplant those cells or the cells derived from those stem cells back into the patient, that they would be very unlikely to be rejected by that person. So your own cells, if we make them from the stem cells, could be transplanted back into you, and your immune system would recognize them as you. And this is what we call, cell therapy for autologous therapy. So back to yourself, as opposed to allogeneic, which is, accepting cells from other sources, not you. So these stem cells are very versatile in that respect, that we can make avatars of you to help us understand a disease, but in other cases, we could potentially put them back into you and they might not be rejected. So at NYSCF, we have developed a set of robotic systems that we call the NYSCF Global Stem Cell Array. And they basically automate this whole process of taking skin cells or blood cells, deriving stem cells, and then propagating them and using them in different disease models and in different ways that we'll talk about. But it's a set of robotic systems that basically automate this whole process and allow us to scale up our work. We can make many hundreds to thousands of stem cells very easily using this robotic system, and we can do so in parallel. So it allows us to scale up to look at populations of people, for example, rather than just one or two stem cell lines at a time in the laboratory. It also has the advantage that it's very reproducible. It does the same thing over and over and over again. We're taking advantage of the robots and automation here to standardize the whole process so that we take away some of the artisanal aspects of biology and creating the stem cell line. So the stem cells that we make from all of the people will have been made in all of the same way, and we can remove a lot of that sort of experimental variability when we're looking at the cells in our different studies. So this allows us to create basically windows into disease. So if we're creating stem cell lines from people with a disease, we can take those stem cells and use them to represent that person in the laboratory and study them in the dish. And we can do this for anybody in any disease. So why do we think that they're the future of medicine then? So basically, we see three really main critical applications of the way that we think stem cells are going to be critical for developing new therapies and treatments. And those are through disease research, trying to understand more about disease and how different diseases affect the cells that they impact. And then the traditional sort of more common idea of basically using those cells to create more cells to replace dead or dying tissues in the body. But they can also be used for drug discovery and trying to find new drugs to treat different types of diseases. And we'll go into each one of these three different applications over the next few minutes. So it's important to remember that most of our major diseases don't really have effective treatments, and certainly most of them don't have cures. So diabetes has been around for generations, and over the past 100 years, insulin has been the therapy to manage diabetes, but it's not a cure. what we would hope for is a therapy that would allow a cure of the disease, to return a patient with diabetes to return their beta cells, the cells that make insulin and respond to glucose back to their healthy state in some way. But other diseases, or cancer patients, don't respond to many standard treatments, and it's really not understood why. Something like diseases like Alzheimer's and Parkinson's disease have very few treatment options available. There's certainly no curative therapies, and there's very few disease-modifying therapies that are available for some of these types of diseases. So there's still a critical need for trying to find new and more effective treatments for all of these diseases that we're interested in. This has really been a challenge in the traditional therapeutic development of different drugs and treatments for all of these diseases. For all of the different drugs that enter clinical development and clinical trials, 80 to 90% of those drugs fail, usually because they have some sort of safety issue, or they just don't work for the disease. They don't have the intended effect on stopping or modifying the disease. And for the ones that do make it, that drug development cost is very long, and it's a long and costly process. And once those drugs do become developed and approved, many of them don't actually work well in all patients. Some of the drugs will only work in a subset of people, and many drugs have side effects that only maybe a small portion of the people that might be affected, but that can be an issue for a very common disease like cardiac diseases that where a large number of people are going to be taking these drugs, and some of the drugs might have potential unwanted side effects. A lot of the drugs are developed using different types of animal models, and in many cases, these animals have not really been predictive of the way that these diseases and the drugs work in humans. So, some of the differences between us and mice, for instance, the mouse heart beats at 500 times per minute, whereas the human heart only beats at 60 times per minute. So, trying to understand how to treat the heart with cell therapies, for example, it's very difficult to do in mouse studies. So there are just some unique aspects of human biology that have limited the ability to develop new successful drugs. We see stem cells as a way to address many of these issues, but fundamentally, we cannot treat what we don't understand. We need to be able to look at actual cells, human cells, that are affected by these different diseases. Many of these cells are not cells that we would really have access to. I would not really be able to ask you for cells from your brain to be able to study different brain diseases. We need another way to do that. And stem cells really offer a path forward here for looking at cells and tissues that we just don't really have access to in the laboratory. And we need access to those tissues to be able to understand how they're affected by different diseases. So stem cells are our windows into disease. So if we create stem cells from your skin or from your blood, create those stem cells and be able to study specific cells affected by disease, we're creating the window in the laboratory into your disease, into your own disease. And that's largely because we're capturing your individual genetics. When we create these stem cell lines, they come from you. They reflect your genetic makeup and the impact of your genetics on your disease and susceptibility to different diseases and treatments and side effects. So we can use these stem cell models as a way of modeling you in a laboratory. So a good example that we've developed over the past decade or more is in Alzheimer's disease. So Alzheimer's is a disease of the brain. Over time, specific areas of the brain and large parts of the brain degenerate. The neurons in that brain tissue start to die off. And it's been noted over time that these plaques that are made up of amyloid and the tangles that are protein aggregates inside the neurons are associated with disease. And a lot of the therapies have been drug development therapies have been directed around trying to remove plaque and tangles from the brain for a variety of reasons and good genetic reasons as well. But the cause of the disease, the actual fundamental cause of the disease, still remains mysterious. So we can find amyloid buildup can be absence in patients that actually present with Alzheimer's disease and vice versa. Sometimes we find plaque and tangles in patients that never have developed clinical Alzheimer's disease. So this disease is turning out to be very complex with many genetic factors and different subtypes. So we have been employing stem cells as a tool to try to understand how our differing genetics can result in the degeneration and the different aspects of Alzheimer's that we see. And we can do that by studying stem cell derived tissues from patients with Alzheimer's in the laboratory. And it's critical because we can now make all the different types of brain cells from the stem cells. So we can make the neurons. The neurons are the communicators that our brain uses to transmit signals. Oligodendrocytes are insulators. They basically wrap the neurons and help them transmit those signals. But then there's other cell types like astrocytes and microglia that they survey the brain and respond to damage or different aspects of brain biology that make them sort of unique cells that we think are very, very critical when we're trying to understand neurodegenerative diseases like Alzheimer's and Parkinson's and many of the other degenerative diseases. So the neurons, we can study those in the dish. They're the communicators. Some aspects of Alzheimer's disease seem to be directly affecting the neurons, whereas other risk factors and genes that we think are involved in the development of Alzheimer's disease impact some of the other cell types in the brain, like oligodendrocytes here, the brain's insulators, or astrocytes, which are the brain's waste collectors. They sort of sense and get rid of different types of debris in the brain. And then the cell's microglia, the brain's microglia, these are sort of the immune type cells in the brain. They're patrollers. They act as sensors to alert the immune system and other cells to damage. And all of these cells work together. And we think that the way that they work together are very critical for how Alzheimer's develops. And we're trying to understand that better in the laboratory. Now we can create more complicated models of these brain tissue in the laboratory using organoids, which are basically taking all of these different cell types and combining them together into little pieces of tissue that we can use to study how these cells communicate with each other and what might go wrong to lead to the development of Alzheimer's disease in the laboratory. Now, so the overall sort of approach that we're taking is to take people without the disease that we know don't have Alzheimer's and people that we know do have Alzheimer's and have been diagnosed with Alzheimer's, and try to understand what are the differences in their brain cell types that we derive from their stem cells. What's the first thing that we can see go wrong? What's the first difference that we can find between the healthy brain cells and the brain cells in the patient with Alzheimer's disease? And you might think that is that trying to study Alzheimer's disease in a dish is challenging because Alzheimer's is a disease of aging. Most people start to develop Alzheimer's in their late 60s, 50s, 60s, 70s. There's rare cases that develop earlier than that, but it's really disease of aging. So we wanted to know and have confidence that the cells that we're making in the laboratory actually reflect the biology of Alzheimer's as it exists in the brains of people who actually have Alzheimer's disease. So as Susan mentioned, we have had this really great collaboration with David Bennett at Rush University, who oversees two different studies called the ROS and the MAP studies. These are the Religious Orders Studies, and the Memory and Aging Project is the acronym. But essentially, these are individuals that are participated in this study. In these studies, have given blood samples, different types of, cerebral spinal fluid samples, other types of biospecimens every year for, in some cases, a couple of decades. And then they also agreed to, at time of death, donate their brain to the study. And this has been a really powerful resource for understanding how our differing genetics leads to our risk for developing Alzheimer's disease. But in our case, in our collaboration, along with Tracy Young-Pierce at Brigham and Women's and at Harvard, we have developed stem cell lines from, in this case, about 53 participants from this ROS-MAP cohort. And we've generated stem cell lines from people both with and without Alzheimer's disease that we know, we know what their course of disease looked like, and we have their brains. And remarkably, the brain cell types that we can study from these individuals have characteristics that match up to what we actually see in those people's brains. So the cells that we have developed in the laboratory by a number of different measures actually do reflect the Alzheimer's brain. So we have a lot of confidence that the disease process that we're actually looking at in the laboratory is actually reflective of real disease biology in the brain. So this gives us a lot of confidence to build going forward, that we're actually looking at real biology here and trying to understand how this disease works. So we have an ongoing collaboration to try to tease out and understand different aspects of the way different cell types in Alzheimer's patients' brains work relative to other diseases. But we're taking this same approach for a number of different diseases. So Alzheimer's is sort of an example, but we're also doing this for Parkinson's disease, for multiple sclerosis, different psychiatric disorders like autism, schizophrenia, post-traumatic stress disorder. The brain cell types that we're able to make from patients with these different diseases, we're trying to understand, what's the first thing that goes wrong in those diseases in the brain cells that we can study in the laboratory. And that'll give us a handle into understanding the biology of the disease. But we're also looking at other diseases like diabetes, type 1 and type 2 diabetes, and these are sort of representative autoimmune diseases. We'll talk a little bit later about eye diseases that we're studying, but also rare diseases that only affect a handful of people, but have very severe consequences for those individuals. But they're also instructive for studying some of the more common diseases and understanding how the biology of rare diseases relates to the more common diseases. And then finally, all of these approaches are really applicable also to different cancers. And we're really interested in trying to understand how ovarian cancer works using much of the same techniques that we use to study these other diseases. So once we've sort of identified how a disease process works in those stem cell derived cell types, that naturally leads us to a system where we can look for new drugs that might revert those different disease signals back to something that looks more healthy. So that gives us a natural platform to do drug discovery. Can we find drugs that actually reverse the disease aspects that we can see in the cell types that we derive from the stem cells? So we can test drugs on specific cell types affected by the particular disease. I think a really good example of how far this has come over the past decade is in ALS. ALS was one of the first diseases to be modeled in 2008, I believe, and using these induced pluripotent stem cells. And we can make the motor neurons that are the type of cell that degenerates in Lou Gehrig's disease in ALS. Over time, Kevin Egan's lab and many others that have collaborated with him and throughout the years, and then finally here with Brian Wenger, who's a NYSCF Robertson investigator, have been looking at using the motor neurons derived from stem cells from patients with ALS to see if they could understand what might be going wrong in those neurons. And they noticed that a particular characteristic of motor neurons derived from ALS patients was that they're hyperactive, they're overactive. And so they did a drug screen to look at initially existing drugs and found this ezogabine. It goes by different names, but it's a drug that has historically been used to treat epilepsy. And they've performed clinical trials to show that indeed in patients that this drug actually could quell the overactive motor neurons, in a key hallmark of ALS. And they're pursuing the development of this drug, but also others that they hope will improve the outcome for people with ALS. But it's a good example of the approach that we want to take with all of these different diseases. We can identify what goes wrong in the cells that are affected by disease, and maybe we can find drugs that actually can treat those diseases. And the flip side of that is, we hope we can find drugs that work to treat the disease, but we also want to find safe drugs. So one of the major reasons why drugs fail is that they're toxic to different cells in the body. We can make things like cardiomyocytes from the stem cells, and one of the main toxicities to many drugs is they affect the heartbeat. The cells that we make from the cardiomyocytes, the heart cells that we make from the stem cells from a person, actually beat and reflect the biology of that person. So we can know ahead of time whether or not that person might have a toxic reaction to one of the drugs that we develop. And this is true for other tissues as well, liver, and there are many groups around the world that are trying to predict the risk of drugs on stem cell derived cell types. At NYSCF, we're also trying to scale up this drug discovery using different approaches like artificial intelligence to try to find sort of key features that may not be so obvious to us if we're just looking at the cells in the dish. We're using artificial intelligence to help us find key disease characteristics and then use those systems to screen for drugs that might make the patient cells look more like healthy cells, as a way of sort of accelerating and improving the drug discovery process. So that's how we understand disease and how diseases work. The other way that we're interested in using some stem cells is basically creating new cells to replace dead or dying or malfunctioning cells that might occur in different types of disease. So if we think about Parkinson's disease, Parkinson's is the degeneration of a particular type of neuron in the brain. There's different clinical studies that are ongoing to try to replace those cells in the brain. Heart disease, it might be possible to replace heart cells in damaged heart. And we're involved in a study and developing a cell therapy for an eye disease called age-related macular degeneration. This is one of the leading causes of irreversible blindness. And essentially what happens is that there's a type of cell in the back of the eye called the retinal pigmented epithelial cells. These are sort of darkly pigmented cells that line the back of the eye and support all of the neurons that are the photoreceptors that actually sense light. So when these support cells die, they cause damage and death of the photoreceptors that are on top of them. And that results in vision loss. And it starts sort of in the center of your field of vision and sort of expands outward as the disease progresses. So what we're trying to do is see if we can replace those dead and dying retinal pigmented epithelial cells, the RPE cells, in patients with macular degeneration. So we can make these cells from stem cells in the lab, and this is kind of what they look like. We've stained the cells with different dyes to show different parts of the cells, but these are, this is probably like 200 or 300, 500 different cells, retinal pigmented epithelial cells, and we can make sheets of these our approach will be to try to transplant these sheets of cells into the damaged area of the eye using surgical techniques that have been developed to do this. So at NYSCF, we're developing a cell therapy to replace these cells that die in macular degeneration. When we've built a facility to make cells that are safe to put into people. So in a clinically compliant, it's called a GMP manufacturing facility that allows us to make the cells that are safe to put into a person. They've been properly tracked and made to a quality that they could be transplanted into an individual. And our sort of inspiration for this study is based off of a somewhat similar type of study that was run by Pete Coffey and his colleagues in London, where they started from embryonic stem cells to derive these retinal pigmented epithelial cells and use those in patches to transplant into 2 individuals in safety trials. And in both of those individuals, they were able to actually restore some of their affected sight in the eye that was transplanted. So they went from reading very few words per minute to reading 60 to 80 words per minute, essentially restoring their ability to read from that eye. There's aspects of the way that of this approach that we think we are improving on by using induced pluripotent stem cells from the individual with the disease that will help us avoid some of the immunosuppression needs that might be needed going forward. So our approach here is to generate personalized stem cells, to make the stem cells from the people affected with the disease, and then make their own retinal pigmented epithelial cells. We put them on little patches that can then be surgically implanted into the back of the eye. And we've launched our facility. I think this is from last summer, and our facility is up and running, and we are generating cell lines for different aspects of towards this project. But we're approaching, we're making our way towards a launch of this study, and we're working with some renowned clinicians, Stanley Chang and Stephen Sang and their colleagues at Columbia University to develop this trial. But we're really excited about the possibility of being able to use stem cells to actually treat and correct blindness, potentially in the long run. But we're interested in basically realizing the promise of precision medicine for everyone. We have a major effort and a high priority to make sure that the research that we do using stem cells will impact everybody and benefit everybody. So we are excited to be able to develop this our bank of stem cell lines that reflects ethnically diverse population. And we have ongoing efforts to try to expand and make sure that we include everybody in our biobank and make sure that they're included in the research that we're doing so that we can benefit everybody. So I hope that I've given you a flavor of what we've done at NYSCF to use stem cells in different ways to both study and treat your disease with your cells. So we really see this personalization as the future of medicine, and stem cells are a key component of how we get towards personalized medicine. So I will sort of stop there and thank everybody at NYSCF, and I'd like to just take a quick moment to acknowledge and memorialize our friend Reed here in the middle of this picture. This was taken a few years ago at one of our last galas, but Reed was a key engineer in developing all of the automation that we have at NYSCF. But more than that, he was a good colleague and close friend to many of us, and we will miss him. So With that, I'll end, and we'll take some questions.
Thanks, Scott. Thank you, everybody, for submitting your questions in advance and during the session today. We're going to get to as many of them as possible. We're going to go about 5 minutes over. We're going to go till about 2:50. So we'll do about 10 or so minutes of questions. So, Scott, you talked about cell therapies, in the work we're doing in age-related macular degeneration. Can you talk a little bit about sort of what stem cells therapies are approved right now, what's in clinical trials? There was a few questions around some specific disease areas like Parkinson's and diabetes and age-related macular degeneration. You talked about age-related macular degeneration. Are there opportunities for other forms of eye diseases like myopic macular degeneration? I know there's a lot there, but maybe you can sort of touch on that.
Yeah, so the type of cell therapy that we're talking about here starts from these iPS cells, and the approach that we're taking for macular degeneration is very similar to the approach that many other groups are taking for other diseases. So most of the first diseases to try to approach replacing different cells that die in the diseases are in diseases where there's a really specific known cell type that actually degenerates. So Parkinson's, I think, is a really good example of this, where just of a small little handful of cluster of cells are actually the ones that degenerate in Parkinson's disease. And there's several clinical trials that are either in very early stages of starting or getting underway as we speak, to try to see if we can use the stem cells to treat Parkinson's disease is a good example. Age-related macular degeneration, we're trying to develop these little small patches of about 100,000 cells to transplant into the brain. The first sort of cell therapies that we're seeing using this technology are ones that where there's a small defined population of cells that are degenerating. But there's hopes that we can improve this and improve our approaches so that we would be able to treat other diseases where larger pieces of tissue might be needed for replacement. So heart cells, bone cells. There are some challenges to being able to create large pieces of tissue like that, but there's really interesting and exciting developments along those lines as well. So I'm expecting over the next 5 to 10 years to start to see some of those start to make their way into the clinical trials and into patients. So, I'd say, for every disease in which a particular type of cell degenerates, there's probably a stem cell approach to seeing if we can replace them. So diabetes is a really good example. So being able to create the pancreatic beta cells, the islets that degenerate in a patient with type 1 diabetes, being able to replace those, but also be able to protect them so that they don't get attacked again by that patient's immune system. So there's lots of different efforts and diseases to try to replace dead and dying tissues.
And you sort of touched on a few things there, and we have a couple questions around the biggest obstacles to actually achieving more cell therapies. And is it sort of the defined populations? Is it the autoimmunity? What are the biggest obstacles to really getting more of these cell therapies into clinical trials?
Yeah, I think that there's several hurdles that we're trying to overcome, and that the field in general is trying to overcome. One is being able to generate larger pieces of tissue, or more complex pieces of tissue in some of the different cases. We've started with very well-defined cell types in cases where smaller pieces of tissue would be used for the actual transplantation. And so the ability to scale up to larger tissues, more complex tissues, is basically an ongoing effort around the world. but also being able to protect those tissues once you transplant them back into somebody with the disease. So being able to scale this to everybody is I think one of the biggest challenges. And we're trying to develop automation to be able to help improve this in different ways. But being able to scale both to larger numbers of people as well as larger pieces and complex tissues, I think those are probably two of the big key hurdles that we're aiming to work our way towards and over in the coming years. But I think there's lots of good efforts towards doing that. So very optimistic. Yeah.
Great. So you talked a little bit about, just right there, you talked about sort of personalizing therapies, precision medicine you talked about in the talk. How are like iPS cells going to actually be operationalized? Can you talk about sort of like both for cell therapy and drug discovery, like are we going to have our own stem cell lines? Are they going to test drugs on our own cells? What is precision medicine going to look like in the future based on stem cells? Or how can you imagine, what can you imagine, it looking like?
So right now we're in the branch of application where we're trying to understand disease. What we're trying to do is scale up to larger populations of people to try to understand how our differing genetics impacts our susceptibility to disease. In many cases, to do that, we need hundreds to thousands of cell lines from hundreds of thousands of patients, or hundreds to thousands of patients, to be able to tease apart how genetics impacts different characteristics of the disease. In other cases, we don't need quite as many, but the fact that we can take those from each individual means that we can, access populations to better understand these types of diseases. But for the cell therapy side, having your own stem cell line, I think, is something that will probably happen in the future, because that cell line could be developed into a tissue that you might need for replacement. But there's other strategies as well, trying to basically hide the cells that we're trying to transplant from the immune system. There are other efforts that are aimed at trying to cloak cells from another person so that we can transplant those into a larger number of people with different diseases. But to do that, we need to hide them from the immune system so that they won't be rejected, like in the case of a transplantation into somebody that would then need immune suppression. So that's something that we would like to be able to accomplish or overcome in the future. Our macular degeneration trial, though, is basically using cells from the patient that we're treating. So the hope is that their own cells would not be rejected when we put them back into the individual. And we'll be looking for that and testing that as we go forward in the clinical studies.
So to change it a little bit, a couple questions came in around cancer. Some different types of cancer, but that people are asking about. So how can this sort of work be used to study and find better treatments for cancer? And can you talk about the work that's being done at NYSCF?
Yeah. So cancer has a cancer cell has a lot of similar aspects to a stem cell. It's basically a cell that's gone out of control and basically replicating itself. And we found that many of the methods and techniques that we use in the laboratory to study stem cells can be applied to cancer cells, to tumor cells. So we have an active program to try to understand and find better ways of treating ovarian cancer. And we're doing this using these little organoids. So taking the tumor tissue, developing it into a organoid, a complex sort of cancer tissue that we can manipulate and study in the laboratory, and that can be renewed and used for assays. And our hope is that we can find new drugs, new therapeutics that would specifically kill off an individual's cancer. So trying to tailor cancer treatment to the individual tumor and the individual's tumor. So a lot of the same ideas and approaches that we're using to study different diseases, we're also applying to, in our case, ovarian cancer, but could be applied to other cancers as well. These organoids actually do a pretty good job of replicating different types of tissue, and cancer is a tissue that we can replicate in the laboratory and use as a tool to study.
So I'm gonna have probably 2 more questions, 2 more minutes. We, when you're making these brain organoids that you talked about, you talked about you studying them for like Alzheimer's disease. I think we can use it for studying diseases like autism and other brain diseases. How are you able to sort of mature these cells, and how do you know that they're representative of the adult cells in the human brain?
Yeah, I think we do a lot of work to try to compare the cells that we make in the laboratory to the actual cells in different individuals using different experimental techniques. But I think that the example that the best example that we know that we have so far is that the brain cells that we make from patients with Alzheimer's disease look like the brain and have characteristics of the brain cells from the individual from whom they were made. And so that gives us a lot of confidence that we're actually looking at real biology in the laboratory. The cells that we make from stem cells are often they can be immature, but we found different techniques from improving their maturity to make them look more and more like more adult type tissues over time. This is important for studying different age-related diseases, but the stem cells have also been really useful for studying, early diseases as well, diseases that affect development and understanding how developmental processes might lead to disease.
Great. So it's 2:50. I'll ask one more question. What excites you the most about stem cell research, over the next couple of years?
I'm really excited about the ongoing therapeutic work that's actually starting to come to fruition. So I gave the example of the ALS trial, looking at a particular drug and repurposing a drug to treat a disease. So I think we're going to start seeing more of this come about, finding drugs using stem cells as a tool. And I'm really excited about the prospects of that over the next few years.
Thanks, Scott. So thank you everybody for coming. There was a question in the chat about participating in research. So my colleague Rekha just dropped a link in the chat for anyone who's interested in participating in research. Specifically, the question was about Parkinson's, and that is an area that we're certainly enrolling patients in our clinical program. So definitely reach out, email us, and hopefully you can participate in the research. So we have upcoming webinars in a couple weeks on AI for brain diseases, as well as women's reproductive cancers in March. And so we hope to see you there. So stay tuned. Thanks, everyone.
Thank you.